Hello, welcome everybody to our April 2022 webinar series. This is webinar 2 in our webinar series, and it's going to be how to restore soil health. So before we get started and let a lot of folks join, if you can, in the chat button, which is going to be on the lower part of your Zoom window, just put in there where you're coming from. We like to know from where in the world is our audience participating from. And yes, the crowd is already responding. We got all kinds of folks in the US. Wow, it's scrolling so fast I can't even read.
Yeah.
We got Netherlands, Ireland,
Nigeria,
Nigeria, folks from Africa.
from Greece, from
Wow.
Seattle. Look at that. Oh, Spain.
Italy, Brazil.
Yeah.
I know. I can't keep up. It's going so fast.
Well, it needs to be global because everybody everywhere needs to learn about this approach and apply it to your specific conditions.
Exactly. Yeah. And it is such a neat thing that we have such a global community.
Bulgaria.
I love it. We have a Skype group that we, for the students and so forth. And that thing goes all 24/7 because it's folks from around the world are always commenting and posting things.
Romania, another England.
Yep. That's
Norway.
great. Got actually a lot of folks from England today.
Yeah.
All right. Well, let's get going because we've got a lot of stuff to cover today. So just some instructions, some webinar rules of engagement. Folks will be muted for the duration of the webinar just to make sure that we get good audio quality. And if you have a question for the panelists, make sure you click on the QA button, separate from the chat button. And I'll explain some of those instructions in just a bit. And then feel free to converse with the other attendees in chat. There's usually a pretty good running dialogue that's going on in the chat button, but also make sure you pay attention to today's topic because there's a lot of good information that we'll be going through. Okay, so I mentioned this is a webinar series. So we've already had webinar 1, which was understanding soil health. So it kind of gets into the science behind the soil food web. Webinar 2, which is today, is going to be how to restore the soil health. So what are the techniques and practices and things that we do to regenerate the soil biology? And then webinar 3, which is going to be a fantastic webinar, we got Ray Archuleta, who's kind of a leader in the regenerative agricultural space. And he's going to be talking about the successes and the transitions that have been made in regenerative agriculture. And then webinar 4 is going to be careers in soil regeneration. So we'll be hearing from folks like myself and Loida, who are consultants out in the field, doing the good work. And so today's topics, we have introductions, so we'll kind of, tee up what we're doing. And then step 1 is going to be finding beneficial microorganisms. And step 2 will be multiplying beneficial organisms. And Elaine will be covering those. And then step 3 will be application to the soil and foliage. And step 4 is going to be protecting the soil ecosystem. And Adam's going to cover those. And then we're leaving some time at the end of the webinar so that we can answer your questions. And we'll have panelists that will be able to take those questions and be able to have a good discussion. We expect that today's webinar is going to be around 2 hours. And also just want to point out that today's topic was actually covered pretty well in depth in our January webinar series. So the topics that we're covering today were discussed in depth in webinar 1, 2, and 3, and you can find those on our YouTube channel. So you can go back and take a look at those. And really, that's still also just scratching the surface. If you really want to get a deep dive into the science and the practical applications behind the soil food web approach, then I definitely recommend that you check out the foundation courses. All right, so today's panelists, of course, we've got Dr. Elaine Ingham, pioneer, founder, incredible woman that she is, really pushing all of us to do the good work out in the field. We've got Dr. Adam Cobb, who's also works at the Soil Food Web School and is an integral part of our content development team and the research that's going on. We've got Loida Vasquez, who's a mentor. She's also a consultant. And then we got myself, who is, just like Loida, I'm a mentor and a consultant, doing the work out in the field. Okay, with that, before we get started and I hand it over to Elaine, we want to ask you folks a question. And it's going to be a quick poll that's going to be basically asking what best describes you. Are you coming from a farmer, rancher, growing kind of perspective? Or are you looking for kind of more of an impactful career, looking to, become a consultant and make those kind of transitive types of work out in the field, moving from conventional to regenerative agriculture? And I think the poll went live. Can anybody confirm for me it did?
Yep, I think you can see the chat at the bottom. Everybody's writing in A or B. Both. There's both.
There we go. I think, and the poll just popped up for me. Yeah. So if you want to click on the pop-up, that'd be great. And typical, I think a lot of people are coming from the farmer, rancher, grower perspective. That's what the results are kind of looking like.
Yeah, home gardener, that goes in that same, so we, of course, will be very interested in those who want to do a career and transition and to helping other people make this transition.
Exactly. Okay, let's move on. And so while we're going through the presentation from Elaine and from Adam, if you have questions, go ahead and click the QA button down at the very bottom, and we will go ahead and pull those questions up for the Q&A session. And if you have just the kind of running chat that you want to talk with others, and that's when you would use the chat button. But just make sure that you're going to ask a question for the panelists, that you're actually placing that in the QA window. Okay, without further ado, Elaine, I hand this over to you. How to restore soil health.
Thank you very much, Brian. Okay, so giving you a little bit of information about how to restore soil health. Of course, we're going to only touch the top of this topic, because when you're thinking about soil health, you've got to understand your own climate. You have to understand what the plant communities are in order to determine what should be out there and then what's missing. So finding those beneficial microorganisms, and I can't advance, so I get to say next, please. There. So where do we find the organisms that we need for soil regeneration? We're going to go out into the local countryside. Hopefully on your farm or in your yard, you've got some trees in there, and so you can be taking the material from below the trees. You just need a pinch or two. You don't need grams worth of things. You don't need cupfuls of things. You just want to take a very small amount and go to the forested area where you've maybe got 2 or 3 trees and it's making nice loamy soil. Go to where you're growing in your vegetable gardens, take some of the soil. So you're seeing what you have out in those different areas. And I would probably put the the forest materials together in a bag, the grassland, your lawn materials in another bag, your vegetable gardens, your underneath your rose bushes, whatever you've got. But we want to get a fair diversity of all these organisms. The further along in succession you go, you're going to get more and more of that complete food web. But we have to be careful not to harm that forest system. You cannot be taking a lot of material from there. Well, what if all your neighbors use pesticides and the pesticides are drifting? Well, you then might want to go into a national forest, or you want to go someplace far away from those toxic chemicals, because those toxic chemicals are killing these organisms. And you need to gather a diversity of starting materials, of starting organisms. And we'll be composting those aerobically, because most of the aerobic organisms are beneficial to your plants. It's the anaerobic microorganisms, those things that grow at lowered oxygen concentrations, those tend to be the disease-causing organisms. That's where we find most of those that are in that reduced oxygen or fully anaerobic conditions in the soil. So we want to be picking up a diversity of different kinds of starting materials. So look at what you've got in your own land. Look at what's, maybe you can go to a local farmer and say, could you just take pinches off the ground? Especially an organic farmer would be very good. So we want to gather the materials from your region. So they are the indigenous microorganisms. We have examples recent of people bringing things over from, from overseas someplace, and, not thinking about what they were doing, brought those plants out into their orchards. And now we've got a disease that we've never seen before. We don't have any natural controls for how do we develop those so we don't keep suffering crop loss. You don't want to be the person that has that mark on them for life. So use your local indigenous organisms. So there's no question that your organisms know how to deal with the bad guys. Okay, so next slide, please. These are the 4 key groups of the soil food web that we want to make certain that we have in your soils, in the compost that you're going to make. And so we are putting these organisms into the compost to make certain that we've got the massive diversity that we need to have in every gram of soil, every quarter teaspoon of soil in your compost pile. We want to be seeing something around 1,000 different species of bacteria. We want to see maybe almost the same species diversity for the fungi. Going to be a little bit lower for the protozoa and significantly lower for the nematodes, but well, they're bigger organisms and they manage more of their property than the small critters. So fungi. And you can see here all of the things that fungi do for your plant. Huge contribution to decomposing organic matter. Really, it's fungi and bacteria that are in competition with each other. The fungi like the very complex food resources. So that's their specialty. Bacteria go after the simple, easy compounds where they don't have to be putting out complex enzymes to be breaking things down. So both the bacteria and fungi build soil structure. If you don't have bacteria in your soil, you are never going to convert the dirt into soil. They are step 1, and that first step has to be managed properly. It's got to get started first, and then the fungi start coming along, and then the predators come along. So we'll show you a diagram of this food web a little bit later, but we focus on fungus, or excuse me, we focus on these functions. So we want to know what each one of these organism groups are doing. And like with the nematodes, you've got to start looking at their mouth parts. You've got to look at their digestive system to have an idea of whether you put them into the bacterial feeding nematodes, the fungal feeding nematodes, the predatory nematodes, nematodes that eat other nematodes, or the root feeding nematodes. Yeah, those are the bad guys. Doom, doom, doom. So how do you select against the root feeding nematodes? Well, there are very specific interactions, and we go through all of that in our foundation courses. Can't get through it all here in this short and brief of a time. So next slide. We'll teach you about all those different interactions and how you control them so that the diseases and pests, the parasites, cannot win. And you will not have those problem organisms attacking your plant. So different organisms need different foods. Just, like think about yourself and your family. Does anybody in your family have exactly the same preferences for food? No, probably not, unless you're a twin. So you need to have all these different food resources. You want all these different organisms functioning under different temperatures, different moistures, different cycles of rain. How hot does it get in the middle of the day? How cold does it get at night? And every time you start shifting that by as little as 5 degrees change in some aspect of temperature, It's a whole different community of microorganisms that have to be functioning. And if you don't have those organisms in your soil, you're not going to be able to grow plants that are healthy. So we want your soil biome, there we got it, the soil biome. If you don't have enough resilience, that's, you know, somebody to take care of the job no matter what the environmental conditions are, you're not going to be able to have the plants that you're trying to grow be 100% on its genetic capacity. Your plant has a certain maximum over which it cannot grow for a number of reasons, but we want to make certain that its ability to reach that maximum level of productivity is very easy for you to accomplish. So every second of every day that your plant is growing, it needs to be getting the balance, not just a lot of nitrate, not just a lot of phosphate or potassium or calcium, whatever. It's all the nutrients. Why do soil scientists not pay any attention to all the other nutrients? Because those can be just as limiting as the macronutrients. So your plant needs everything. How do you make certain that the nutrient cycling is going to supply everything? You've got to have this kind of diversity of the bacteria, of the fungi, the protozoa, and the nematodes at the very least. They have to be balanced properly. So when we're making compost, 3 different types of starting materials. Next slide, please. High nitrogen, green, and brown, or I always say woody. So high in carbon when we're looking at the brown woody. When we're dealing with high nitrogen, you have very narrow ratios of carbon to nitrogens, around 5 carbons for 1 nitrogen. So think of a carbon molecule sitting there. You've got to have at least 1 nitrogen for each 5 carbons. Well, as you get into less and less nitrogen being put down in the plant, as the plant grows, it's going to go from something that it has a C/N ratio of 5:1 or 25 carbons for every 1 nitrogen. As your plant grows, it starts to put on stronger and stronger stems. It starts to put out defensive mechanisms like thorns. So there's a lot of things that are going to have a wider, different parts of your plant that are going to have a wider carbon to nitrogen ratio. So look, now with the green guys, green nutrients, you're looking at C/N ratios 25:60. Who is it that flourishes when we have lots of green stuff? It's your bacteria, because they're much more simple compounds, easier to break down. You only need to be making 1 enzyme at a time. When we move into the brown or woody, high carbon containing materials, so look, the C/N ratio is over 60:1. It could be as high as 500:1, 1,000:1, and that's going to severely limit any microorganism that requires a lot of nitrogen. It's going to limit the growth of those organisms. So high nitrogen, we're putting out party food for our bacteria and fungi. So they'll start growing so fast that they'll start to generate heat. And it is that heat that is going to deal with the disease-causing organisms. Now, it's got to remain aerobic. Remember, we were talking about that before. But the high heat with plenty of oxygen, we're going to wipe out any disease, pest, or parasitic organism that might happen to be in your mix of starting materials. So here's 3 things you've got to keep track of when you're making your compost, when you're making your soil. You need the proper ratios. So we teach you what those recipes are. And please remember, these are general values. The recipe is going to be different for the springtime. So when you start to go out and work on your soil early in the springtime, you may be putting on one kind or one set of communities of bacteria and fungi and protozoa, nematodes, microarthropods, etc. But when the summertime comes along, you realize you have to shift that ratio a little bit. How about in the fall? Everything's going to sleep, large amounts of residues, plant residues going down on the soil surface. And then even in the winter, the most rapid rates of decomposition of organic matter occurs in the winter, under the snow, because there'll be free water at 4 degrees Celsius. You will have enough water in liquid form that these microorganisms, well, and it's going to be the cold loving part of your diversity that you want to have in that wintertime period. So when the snow melts, all of those residues left from harvest at the end of the summer last year, all of those residues have been incorporated into the soil and they are now part of the soil. Think of all the nutrients that are now in your soil because these organisms have done their job. You're going to sequester carbon. You're going to have a much more of your nutrients held in the organic matter instead of having any chance to leach out, any chance to be lost from the system. Next slide. So we need to avoid some kinds of things because Mother Nature has allowed organisms to make compounds that will protect them from being decomposed. So you want to not be putting in things that have a natural barrier to bacterial and fungal growth until that material has been slowly but surely decomposed especially by the fungi in the system. So think about the antimicrobial agents in pine needles or pampas grass or, any one of those. Well, walnuts come to mind. Certain, even alfalfa makes allelopathic compounds. And those have to be dealt with before you put them into your compost. We don't want to be slowing down these processes once you get the compost started. You have to avoid all of the pesticides, insecticides, herbicides, fungicides, nematicides, asides, so that you're not killing the very organisms that you need to have to do these processes for you. It costs you time and money and gasoline, the cost of putting out pesticides, insecticides, etc., or using inorganic fertilizers. I don't use any inorganic fertilizers. There is no need because your soil contains plenty of those nutrients. So, I know some people like to, well, can't we just put out a little bit of inorganic fertilizer because we're really not sure. So usually it takes them 1 to maybe 2 years to go, I was such a fool, and say, I wasted some of my money. Yeah, I just couldn't quite grasp the reality that for all those years they had been farming, they were paying money to people who were not really telling them the whole story. So avoid those toxic chemicals. It's just insane that people think that they can grow good, healthy plants, highly nutrient-full plants by applying poisons. Just doesn't make sense. So we got to come out of that green revolution attitude where they had no idea of what the biology was in the soil. We want to avoid manure from animals that were treated with toxic chemicals, dewormed, or they've been fed formulated feed. If the feed is put into a bag, you know that they've got to add into that material something that will prevent decomposition of that feed or pellet. And so you're putting toxic chemicals, high salt levels, typically, into your ecosystems, and you're killing the very things that you need to not have to do deworming, where you don't have to worry about the quality of the pellets or the feed that has been produced. We don't want you to be taking a lot of organic matter from the natural systems around you. So how long do you have to go out and be collecting those organisms? Well, in the first time that you try to start compost piles, you'll need to go out and get your inoculum. And now you get all that huge mixture of diversity of the bacteria, fungi, protozoa, nematodes, microarthropods, all those critters. And that's, you don't really need to be doing anymore, because that really great compost will attract the other beneficial organisms to that area. And so sooner or later, Mother Nature is going to be taking over that job, which means you get to go put your feet up and not have to worry about it anymore. Next slide. How to restore soil health. And multiplying beneficial, well, no, we're multiplying beneficial nematodes as soon as you start putting anything into the soil, anything that has that diversity of microorganisms. So what we're really trying to say here is not really so much multiplying the beneficial microorganisms. They'll do it themselves with, you don't have to supply that workload. But what we're really looking at here is making sure that we're balancing the bacterial fungal ratio. We're balancing all of the nutrients that are being made properly in the soil and making them available to your plant. So next slide. So what is BioComplete compost? It's got all of the organisms in the soil food web there present in more or less the right balances. It's got to stay aerobic. So look at all the airways and passageways that you want to have in a good, healthy compost or soil. Pick up a handful of it, shake it. You should be able to see all those air passageways, lots of spaces to hold water, to have oxygen move through, have your microorganisms move through, and even more important probably is getting the roots of your plants to grow through. You have to build the structure in all of these materials. And the only organisms that will build structure are the bacteria, then the fungi, then the protozoa, then the nematodes. They all have their role and their function. We want consistent soil levels, moisture levels. When you're composting, you may have to add moisture to that compost pile. And you've got to be measuring the temperature all the time so it doesn't get too hot. You don't want to burst into flames. So just with a modicum of care, you learn the recipe. And so BioComplete means we've got all the organisms at the minimum level. And then we want to look at balance and make certain that the balance of the bacteria and fungi, protozoa, and nematodes is exactly what your plant requires. And how do you figure out what your plant requires? You've got to understand succession. So next slide. Next slide.
Did
okay,
not come up,
so
Elaine? Oh, there we
no,
go.
it
Okay.
finally came up on my computer.
Sure.
It was, we were having a moment of silence.
Yes.
So the objectives, we want to grow the populations of the beneficial organisms, and the control factor that we use is aerobic conditions. There are very few truly devastating disease-causing organisms that can outcompete aerobic organisms. Aerobic organisms, I can't go that deep into, I would love to spend 2 or 3 hours on this. So instead, we will just grow the populations of beneficial microorganisms. And that's why having consultants is so important, that when you're not sure, you're not, oh gosh, what do I do? You either look through your microscope and figure out what's missing, what's wrong, how come this soil isn't forming structure, how come it's not getting fluffy. All of these visual indicators, the color is not what you want. Consultants can be very useful in trying to help you go through point by point by point, what's the most probable problem. And then let's look through the microscope. Yep, and that's the problem. And so in the course of a very short period of time, you figure out what's wrong and you apply what's going to fix it. So we're going to make sure we put in the beneficial or microorganisms that are aerobic, not anaerobes. So I think I've beat this one to death off already. Compost has to be aerobic. If you ferment materials and you allow your compost to go anaerobic, you're doing very different things than what we're trying to do with the compost. You know, you can make pesticides all the time. You have at your own discretion, you can be making things that kill the bad guys. The problem is those materials are going to kill the good guys as well, which ultimately means you've got to go back and you've got to have a good compost. So show a modicum of patience when you've got a little bit of disease problems rearing their ugly heads. It's Mother Nature's way of telling you that you haven't reached the desired balances. You don't have the organisms right, so keep working on it. You're close, but no banana. So you don't ever have to be putting on these homemade pesticides. So next slide. Aerobic composting method. So I've briefly pointed out the things you really want to be looking at. You want to measure these things to make sure that the biology is growing properly. Look at the steam coming up off of that compost pile. And a lot of people would be worried, oh, it's going to burst into flames. No, no, no. When the temperature in the pile is higher than by 15 degrees than the outside ambient temperature, you very well could be getting steam from the pile. That's all it means, is that the temperature inside this pile are growing so fast, they're releasing all this heat, they're killing all the pathogens, and you can see with that steam that the good things are happening. Now, if you stick your hand into the middle of your pile and your gloves start to burst into flames, you better do something to bring that temperature down. So, and we teach you those things as well. Next slide. So typical list, you know, this is to just indicate to you that this is not expensive. We're not making, especially for small scale growers, it's a wooden pallet down on the bottom, so you keep the bottom of your pile out of puddles of water during the springtime. In the late summer, you may take the pallet away because you don't need it anymore. You have the wire cage. I call them my bird cages, compost cages, a suitable water source. So that means you cannot have toxic chemicals in the water. And when you're drinking city water, when you're drinking water that's come through a plant, a clean water plant, you're going to have some toxic materials in that water that supposedly are not harmful at those concentrations to human beings. But it's sure going to do a job on the microorganisms in your digestive system. So, think about that conundrum. You really want to take the chlorine and the chloramine out of your drinking water so that your digestive system can actually function the way it's supposed to function. 5-gallon buckets. How large do you want to make your pile? So you're just going to have to pick up, 20 5-gallon buckets or 40, 20-liter buckets are what we're talking about. For those of you who are not in the only country in the world that's still, never mind. Okay, tarp, a pitchfork, pretty easy. You're going to be turning this by hand, but most people find it enjoyable to get out from behind their computers and have some physical work to do every time you need to make more compost. And, small scale, homeowner size, you're only probably going to be making 1 or 2 compost piles through the whole year. As you go to larger and larger and larger, you just make more and more until you start making windrows. We work with people who have 10,000 hectare growing farms. And so we manage to cover everything. We have the ways to go up to those scales. So where do you want to get into this system and make something that is fun and isn't going to end you up with back pains? Okay, next slide. So,
Okay.
Yep.
I think that concludes part 1 and part 2, Elaine. And I think we're going to take a quick break before we move on to parts 3 and parts 4, just to talk about our Springboard Plus offer that we've got going for our April promotions. And so we've got right now the April promotions running, and the courseware is on sale from $5,000 down to $2,900. So that's 47% off. That's the lowest price that we are going to have through 2022. And there's also going to be some bonus courses that that we're going to mention as well. And to describe all of this and the offer, we've got a short video that we're going to go ahead and run through. So let me go ahead and start up the video. And here we go.
Spring is in the air, and there's never been a better time to launch your career in soil regeneration than right now. With the Springboard Plus offer, you can register for the Soil Food Web Foundation courses for just $2,900, and you'll get 2 free bonus courses, saving an incredible $2,600 or 47% off the regular fee price. This is guaranteed to be the lowest price through the rest of the year. Whether you're a farmer, rancher, market gardener, or just someone who's passionate about the planet looking for a way to make a big impact, this could be for you. Here's what some of our students are saying about the Soil Food Web training program.
I find that this information hasn't been taught to me, and I had to get off my high horse, and even though I have a PhD, I feel like I'm totally undertrained.
I feel like I'm learning more with this program than I have with in-person classes in the past.
I've taken classes on microbiology before, but this course really makes a difference in the way that a story is put together that unveils the relationships between plants and all those beneficial organisms that we just cannot see without a microscope.
If you're looking for something that does a deep dive into soil biology, this is it. It is just an incredible knowledge base and is really relevant to what's going on right now in the world.
Without it, the only way I could have gained this knowledge would have been by dropping my life and going to graduate school. And that just wasn't realistic for me. But Soil Food Web has made it possible for me to build a meaningful career in land restoration.
I was really nervous. I was going to put quite a bit of money down and not get that bang for my buck. But once I actually got into the FC courses, I was incredibly impressed with how professional they are and actually the level of education you receive. This is the career path I've been looking for in the biological community, and I was having trouble finding.
Remember, with the Springboard Plus offer, you're not only getting the foundation courses for $2,900, but you're also getting these 2 free bonus courses. The Introduction to Permaculture is an 18-lecture course that covers a wide array of permaculture principles and themes delivered by Graham Bell, Chair of Permaculture Scotland and longest-serving permaculture teacher in the UK with 31 years experience teaching on 6 continents. Permaculture is a regenerative design approach that can be applied to just about anything, from water management, growing systems, dwellings, and much more. The Soil Sponge Regeneration Workshop is delivered by educator and author Dee Dee Pershaus. This 5-session course is all about regenerating the soil sponge for flood, drought, and wildfire resilience. It builds on the successes of innovative land managers around the world who are saving huge sums in damages from extreme weather events and crop diseases, while restoring the dignity and profitability of farming. Dee Dee teaches participatory workshops both in person and online, helping to show the nested relationships between soil health, human health, water cycles, and climate resiliency. Sign up for these amazing courses and join the soil revolution today.
All right. Oops, I seem to have messed up my screen share here. Sorry, folks. Give me 1 second. Okay. So thank you all for watching our 3-minute promotional video, and we'll also at the end of the webinar just remind everybody about the promotion that we've got going on. But at this point, I'm going to hand this over to Dr. Adam Cobb. So Adam, would you like to take it away on how to restore soil health?
Yeah, thank you very much, Brian. Wow, I feel so lucky today because Dr. Elaine has done the really hard work of expressing a lot of the principles, and I get to talk about some of the success stories and some of the high level, just exciting things out there. And I will mention though in that video, I love that one of the things that was highlighted is the idea of land restoration, because many of these examples that we use often relate to agriculture, and agriculture is something I've been passionate about for 15 years. But these same things are applied as they're discussed in our foundations courses to rehabilitate landscapes, to do ecological restoration of forests, and to be used to set up parks and other things in really less destructive ways. So yeah, if we could take us to the next slide, Brian, I love to talk about Adam York, who was on our January webinar number 3. And it was really great to meet him and hear about Agribio and some of the work he's done with Todd Harrington and other folks that have worked with Elaine in the past. And really, if you look at this case study, I love it because oftentimes in the circles I run in, people are a little skeptical that this could work on a big scale, right? How would you ever make enough compost to fix the soils on a huge farm and all? And Adam York has proved that he's working on something like 45,000 hectares of land, which is like 110-ish thousand acres of land. And just has been moving that system further and further away from any need for chemical inputs and going all biological with that. So if you could take us to the next slide, please, Brian. I just wanted to highlight that that it was a process for Adam. And again, you can hear directly from him in January webinar number 3, but they had to go through, much like our CTP students here at Soil Food Web School, a learning curve. There's a learning curve to this. It's not just a one-size-fits-all solution. The principles are universal, and it's up to each participant, each student, each person trying to apply this in their garden or farm or in landscape restoration to find a system that works for them. Equipment is a huge part of that, and equipment's variable. The local climate and other conditions are all in there, but they made clear and steady progress at York Farm, being able to scale up to where they were producing thousands of gallons, many, many thousands of liters of biologically complete liquid amendments. So if you could take us to the next slide. This is just a snapshot of some of the work that they do. And, it's work. They have to keep on this. There's a lot of production of liquid amendments. So after the composting process that Dr. Elaine explained to us, when we know that we have a good balance of microorganisms from across those key soil food web functional groups, then it's about getting those organisms into an application ready liquid state. This is covered in our foundation course number 3 in detail. And so Adam York and his group, they have taken that up to a big scale, and they're able to produce the compost at a big scale, but also to get that compost into systems where the organisms can be extracted, and in some cases where foods can be added to that liquid amendment at the right time, that they can actually grow out the fungi or bacteria, even protozoa that they're trying to reestablish in those landscapes. So on the next slide, we have one example of a way that they've been able to apply that, which is that kind of typical pivot irrigation system that you see all over the US, the Midwest, whenever I drive through Kansas or Colorado. When you're flying over in a plane, I've had friends ask me, why are all those fields down there in circles, Adam? And it's because there's these center pivot irrigators that are adding irrigation water to the crops. Well, if you do some adjustments to that equipment, details that are covered in our foundation courses, you can take these liquid amendments and you can add them directly to the crops very easily on a large scale. And that coats those, say corn in this case, that coats with beneficial organisms a few times during the year so that you end up armoring the plant against lots of pests and diseases. And then you can also, those sprays will get all the way down to the soil and will keep reinvigorating that soil biology in case something in your management of the farm causes a little harm over time. And so we have another example slide coming up next. This is just where Adam York's group had, they sometimes are transporting out and applying these liquid amendments, in different scales. So pasture systems, for example, are not typically pivot irrigated, but a lot of times there is some kind of spraying equipment that can be adapted. Things that used to spray toxic chemicals onto the landscape can be adapted, cleaned carefully, tested, and they can be, that same kind of equipment can instead be used to spread living soil organisms. And then I think we just have one other example slide here of the kind of tractor-based equipment that can be used to spray as well. Now, there's a lot of details that we can't go in on today about the size of the nozzles may need to be adjusted, the intent, like the pressure of the liquid as it's coming out, because we have to make sure that when the microorganisms land, they aren't instantly destroyed. Right? Because it's maybe coming out at such high pressure. So all of these kinds of things are discussed in foundation course number 3. But it's just exciting for me as someone, I have lots of friends who are in large scale industrial agricultural production, maybe of wheat and cattle. And this helps me overcome some of that skepticism that's there, that Adam York and others like him around the world are figuring out how to do this on a large scale and saving money, because that's what we really want, especially ag producers. It's reducing your input costs and having enough output yield to sell that you make more profit. And so one of my favorite examples with Adam York's case is that he used to spend a lot more money on seed because he was buying genetically modified seed and then spending a lot of money on Roundup to spray on his corn so that it would kill the weeds. And now he's spending much less money on seed. He's investing in different products that can feed microbes instead of Roundup. So instead of killing his soil biology, he's investing in making his soil more resilient, more thriving soil food web each year. And it's just very inspiring to be able to look at this case example. So I hope you all will check out the January webinar 3 to hear more directly from Adam York about his journey through all of that. So I think that'll take us through step 3 here, Brian. And we'll get on to protecting the soil ecosystem, because this is something that comes up a lot. We focus at the Soil Food Web School on getting that soil food web back in the system. That's what we really do well. We'll teach you how to assess the soil food web that you already have in the landscape, and then how to identify what's missing, and then how to grow out amendments that can restore the balance and the diversity and the abundance to that soil food web. And then after you've done that, if you still till your land every year, you're going to keep destroying the soil food web. And so how do we manage, how do we design systems around minimized disturbance to the soil food web. So you'll take us to the next slide. I would like to tell a little story about when I was in grad school. I took this picture in Kansas, in the Flint Hills of Kansas, in one of the few areas of intact tallgrass prairie that still exists in the US, right? And something like 90, some people even say 98% of the tallgrass prairie was tilled up and destroyed and turned into wheat fields. But it's so rocky in the Flint Hills that there were sections left undisturbed. And when I was a graduate student, I kept wondering, how is it that this grassland that has maybe every meter square has 16 plus species of plants, legumes, grasses, beautiful wildflowers, how is it that this system produces so much grass and that it has historically fed something like 40 to 60 million bison with very little management from humans? The indigenous communities were not adding fertilizers. They were not using herbicides. They were sometimes using fire as a management tool on the landscape, but there was very little costly input going into the management of these grasslands. How were they so productive? I didn't have an answer for that question until I came to Soil Food Web School about 7 months ago. And if you'll take us to the next slide, please, Brian. This is the answer. The soil food web is intact in this intact piece of tallgrass prairie. All of these relationships have been allowed to do their emergent natural magic, for lack of a better word, work together in synergy. Human beings have not crushed nature with our boot on its neck in this place on Earth. And so we can look into this system and see that the sunlight is coming into all of this diverse plant community, and it's accumulating diverse kinds of organic matter, diverse exudates from the root systems that's feeding tons of bacteria and fungi. Those bacteria and fungi are absorbing nutrients from the organic matter, pulling it from the rocks. Those things are in turn being eaten by protozoa and nematodes so that the nutrients are released. There's arthropods, there's earthworms, there's things that are just active in that soil system. And it's this beautiful symphony, really, of organisms. So if we have the presence of all of these components of the soil food web, and if they're abundant and thriving, and if we see diversity there, and if there's balance between the different predator and prey groups, for example, then you end up not needing to add a bunch of costly inputs, and you can see massive productivity and resilience. These systems periodically faced drought, and the plant communities could still produce in a drought year, right? Whereas if you talk to a industrial corn farmer, drought will kill their yield. You know, they'll be like, I'm going to go bankrupt. I'm not going to pay the banknote this year because of one drought year. So resilience is the word that keeps coming to mind when we're talking about a thriving soil food web. If you could take us to the next slide, please, Brian. This is a graphic that I came up with recently when I was going to a conference in Greece, and I thought about the arch and how an agroecosystem can be thought of like an arch. There's all these different things that we're trying to manage in an agroecosystem. What kind of crop we're growing, the timing of when we're doing different practices, how that food production system comes together economically, what kinds of breeding are happening with our crops, how we're managing the habitats and the stress and all of that. And for a long time, nobody was talking about the center of the arch, the keystone, until we had this emergence of the idea of the soil food web. And you can't hold together a sustainable agroecosystem if your soil food web is missing. It's the keystone. it's holding together everything else that you're doing. And so when there's all this research and development in agriculture that focuses just on breeding crops or just on irrigation management or just on some kind of mechanical piece, but they ignore the soil food web, then they're making an arch that's going to fall down. It's not going to last. We need this holistic and scientific approach. And so this was the answer that I got when I came to the Soil Food Web School, and I took the foundation courses and started training with Elaine. Is what I've been seeing in the world about farm systems that collapse because they're fragile. It's because so many people are farming on soil that is a destroyed ecosystem. There's a few bacteria and some pathogenic fungi, and everything else is missing, right? Because of the historic damage that we've done. So I think I have one more slide here, Brian. The larger discussion that's been going on in regenerative agriculture often ranges around these practices that we would do in a farm system. And it's worth pointing out every time we talk about regenerative agriculture, it's not new. These practices have been used for millennia, especially by indigenous populations around the world, because their food system wouldn't have supported their population if they weren't using practices such as this, minimizing their soil disturbance, bringing in a diversity of crops, especially things like legumes that can restore nitrogen to the system, keeping the soil covered with mulch or other things so that it's protected, growing something all year so that there's always sunlight coming into some plant, turning into sugars and other important compounds and going of roots to feed the microorganisms, and integrating livestock into that system. These are all such important practices for farmers to utilize, for farmers and ranchers to utilize. But again, if we don't restore that soil food web, then all of these beautiful practices that are going on are missing that keystone element. They're not going to be as useful. And so, as you learn about permaculture or some other, regenerative techniques that are out there, I just hope that you'll remember that the soil food web is the glue that's holding all that together. Anything about designing better farm processes or managing agri-ecosystems more effectively, if we're not assessing and correcting imbalances in the soil food web, then we're missing the center of the whole thing. So, I do think I have one more slide, Brian. It's one of my favorite quotes, because back in the 30s in the US, this brilliant ecologist and farmer named Aldo Leopold looked out at the way that farming was being done in the 30s in the US and said, our relationship to the land is broken. Our hubris as humans, we're ignoring the way that nature works. And so he said, I'm very concerned with how people treat each other, but I also want to change people's relationship to the land. And I think that that is the ultimate goal of spreading regenerative techniques, of teaching people about the soil food web, is that we humans would approach nature with a different attitude. That we would come in and say, nature, you're so wise, please teach us. What have you been doing for millennia? How can we follow those processes and understand those relationships and work with them instead of imposing our own damaging systems of tilling the land and cultivating the way that just because our grandfather did it that way, right? So that's the vision that I'd like to share as part of managing the soil ecosystem. Yeah.
Great words to live by, right? Okay, we're going to now move into our Q&A section. I always love the Q&A. It's a great way for us to be able to take questions from you and the audience and have a good discussion. And hopefully you guys will enjoy it as well. So let me just make sure. I think I've got the questions. Oh, I don't see them in my slide deck. So I will just go ahead and read off the questions, and then we'll have the panelists go ahead and answer. So give me 1 second while I pull this over. The first question is from John. And John asks, can municipal compost facilities produce biocomplete compost when using biosolids? What are the challenges? My local facility describes their product as ground yard trimmings, and biosolids are mixed in a 3-to-1 ratio and composted in aerated static piles. Okay, Adam, Elaine, any comments here?
I think they could make decent biologically complete compost. The problem is that they're really more concerned with waste reduction. And so when they get around to the making piles, they're just trying to get those piles to shrink from, maybe 20 feet tall down to 2 feet tall, because now that stuff can go in the landfill. Well, it's a really poor choice in terms of, elevated CO2 in the atmosphere, plus some other nasty things like methane and nitrous oxide and things like that. It's just not done properly. The rules for their processes are not likely to allow a decent real compost to occur, where we really need to understand that compost is the heat is used to kill the diseases and pests and pathogens, not try to blow more gases off and have things escape into the atmosphere. We want to, disease free, but we then want all those beneficial organisms to still be in that pile. There is just no way that you could have all of the beneficial organisms still survive the composting cycles that they go through. It's not possible. They're not going to be there. You're going to have bacteria, maybe some disease causing fungi, actinobacteria, but bacteria out the gazoo, and you're only going to be able to grow weeds.
Yeah, I'm starting to do a lot of work with some of the large scale composters and some of the municipal facilities. And really it comes down to, I think, just a lack of knowledge on their standpoint. Their primary goal is waste reduction. Material they get in, waste reduction, and then, hey, I got an output that I can now potentially sell or give away to the populace. And some of them are starting to really understand that, okay, there's another role that I can play in this, which is now I can create a product that actually has good biology, can be used in regenerative agriculture, and they're starting to make those changes. And like you said, Elaine, it's the processes they use. They've got material. All we need to do is just change the way that they're actually using those processes and treating that material to make good biologically complete compost. And I think for us, especially the work that we do, one of the limiting factors that I always run into is I have a client in an area that doesn't have anybody that's making good compost. It's tough for some of those producers to say, oh, well, now do I have to make on-farm compost? They have to go through a whole process. If we can get a lot of the composters to start making good biologically complete compost, this transition to regenerative agriculture and the use of the soil food web will happen much, much faster. And it's getting there. I think there is an awareness that's starting to happen in the composting world about, oh, there is something I can do to make a higher value product. So, yeah.
That's great. I would mention the word biosolids. To me, I think that has been defined to me as the solid material left over from wastewater treatment, which includes a lot of human waste typically. And I do get concerned about heavy metals, even pharmaceutical residues being in that, and as well as pathogens. But I remember reading some papers in grad school out of Australia where they were using vermicomposting to turn human biosolids into a safe product at the end, that the worms were basically detoxifying, or the organisms that the worms were spreading were helping to detoxify and lock up, which is something that Dr. Elaine teaches that fungi do as well, that they can grab a heavy metal that would be dangerous and put a bunch of other compounds around that lock it in place so that it won't be doing damage. So there's a lot of cool research that can probably be done with closing the loop on our nutrients as humans so that they can go back into our agricultural systems.
And we have to get the regulatory agencies understanding that what they've been labeling as compost is not in any way compost. It's not how they used to grow plants. You know, the father of genetics grew those pea plants using actual compost instead of reduced waste. So, you know, it's been with us for a long time, really good compost. It wasn't until the Green Revolution started to happen that people turned it into a waste reduction process, trying to get the amount of material going into the landfill to be much less in volume. But then that means all that release of greenhouse gases to the atmosphere. So where was the real problem in getting all those gases into the atmosphere? Where did it really come from? Well, I think the waste reduction mindset had a huge amount of effect in increasing that those gases that cause heating. And that's a real problem with those gases that hold the heat in so that it doesn't radiate out into the universe.
And I think there's one other layer to this too, is that our regulatory boards, you know, that are regulating the composting industry also have to become aware of their role in helping also move from that waste reduction mentality into, now let's make a product that can be very beneficial and used to help restore soil. And I know that there are some regulatory boards that are starting to get to that point where they're really starting to understand the science behind it and starting to move in that direction, but we just need more movement. So, okay. Any other comments from the panelists before we move on to the next question? Okay. Let's move on to our next QA. And this question is from Mulugeta. Hopefully I pronounced your name correctly. But the question, is there a standard methodology / approach for correlating the diversity and operational taxonomic units of microorganisms in crop rhizosphere, notably bacteria and fungi? What say you, Elaine and Adam?
Yep, there is a simple method, something that we teach to average everyday people every day. Doesn't take that long. So using a microscope, we want to have it be a shadowing microscope. So you've got to have a condenser and an iris diaphragm below the stage where your sample goes. And trust me, you'll understand what that means in about 10 seconds when we're teaching you what that means. So we want to be able to look at these organisms and determine, do we have the organisms that we require? Now, the tough part gets to be looking at function. We look at the mouth parts, we look at the kind of nutrient cycling that is being done, what's the glue layer. So we look at a lot of those functional characteristics and not have to go to species. We don't have to go to genus level when we're trying to just get a good idea of what's going on. You know, when we look at our at the number of microorganisms, for example, bacteria or fungi, the number of bacteria that have actually been identified, and we know exactly what they do, and we can grow them in the laboratory. There's 5,000 of those microorganisms that we have identified in that fashion, so we could do studies on them, diversity and things like that. But then think about the fact that in a quarter teaspoon of good soil, good compost, has the necessary organisms to grow old growth trees, for example. Minimum, you have 1,000 species in a quarter teaspoon. And the next quarter teaspoon has almost completely different sets of organisms, and the next teaspoon, and the next, and the next. How many microorganism species exist on this planet? We don't have any idea. We're barely touching that process, understanding what what's going on. So yeah, I think we have to focus on function and on biomass, on how many of these aerobic organisms are doing the jobs, and we can perhaps use different nucleic acid sequences to help us identify functions, but there's just a boatload of work that has yet to be done in this area.
Yeah. And I agree that the question around standardization is a huge issue. We have to have a standard working set of data that we can then interpret and then decide how the things that we need to do to remediate that soil or that property. And I really appreciate the work, Elaine, that you did to create those standards. Now that we have a lot of laboratories that have sprouted up to do that kind of measurement. I feel confident that when I get a soil report, a microbiology assessment from one of my clients that was done from a lab that was trained on the Soil Food Web School, that
it is certified.
It's certified, exactly. I know, like, okay, that the data I'm seeing is trustworthy. I can trust that. And as the biological approach to farming is rapidly increasing, there are a lot of other players that are coming in board. And so, it's sometimes hard to trust, have they gone through that rigor and so forth. So I agree that some kind of standardization is going to be really important for us moving forward, that we have a way to be able to assess the microbiology and then have a good way to be able to interpret that from there.
Yeah, and even in the I don't purport to be like a real expert in the molecular genetics world, but I know enough to know how little is known. I've been at conferences where senior researchers are basically yelling at each other about methodology, and there is some resolution that's happened, like in my field with mycorrhizal fungi in the last 5 years, where we're getting some idea about diversity and looking at things like what environmental filters cause the taxonomic units to cluster versus spread out and all in the data. But I asked a friend who's like much more of a fungi expert than me, or much more of a genetics expert than me. I said, how many phyla are there in the kingdom fungi? Because I'm getting different things online. And he says, well, people are arguing about whether it's 7 or 15. And I was like,
small range.
That's a big range for, like, phylum is not even a very specific kind of taxonomy. So it's just, it continues to be, science always, moves forward, and we're keeping our eyes on that here at the school. But I don't think that the molecular genetics are there yet to really answer everything either. So I appreciate the microscopy that we train here as being very practical.
Mm-hmm. Agreed. Anything else for the panelists before I move on? Okay, let's go to the next question. The next question is from Carolyn, and I like this question because this is waste materials that we have to deal with. Is there value in adding wood chipped material from trees in the area that were in a forest fires?
Absolutely. All that good carbon to nitrogen ratio, we need to have the wood chips as part of the diversity. So we're going to typically say no more than 30% of your pile should be wood chip material. You want other kinds of food resources for other species of bacteria and fungi. Probably more important for fungi to have that chipped wood material. But you want other kinds of woody material, wide C to N ratio materials in your pile or in your soil as well. When a plant, when there's a strong wind and you lose a lot of branches and lots of bits and pieces of wood flying by, land on the surface of the soil, you want to have somebody in your soil that's going to decompose that and turn it into something beneficial, not something that's going to cause more disease. If you don't have anybody home and you're trying to grow things with dirt, see, dirt requires both the foods to feed the organisms and all of the organisms, so nutrient cycling is happening. So if you don't have those beneficial organisms in your soil, then you're going to have the disease-causing organisms growing on those foods. It's not like the foods aren't going to appear. They're gonna. But if you don't have the organisms, if you've been killing them, yeah, you're just going to have worse and worse and worse diseases and pests and problems. Good luck.
Yeah, and the reality is that as our climate is changing, forest fires are becoming a much more prevalent type of thing, and there's a lot of material out there that we can then really transform into compost that we can help restore a lot of that land, both back into the forest. I mean, there's that forest restoration aspect too, taking the materials that are, unfortunately left over from that forest fire and helping those ecosystems recover.
And then think about all that partially charcoalized biochar
Really,
that's
yes.
out in those systems. The fire that burns so deeply into the soil, where most of that wood is no longer recognizable as what tree it came from. That charcoal material is a good resource to grow some really great fungi. And we need to re-inoculate because the fires were so intense and so high temperature that the soil was basically sterilized. There is no life in it left. We, being responsible members of this planet, need to go out there and apply the inoculum of all these different species. We don't know which species are going to be growing in which place. So just get them all out there. Maximum diversity, just get that diversity out there, and the microorganisms will split up the foods and the resources, and you'll have soil in a very short period of time.
Adam, any comments? You good to go?
Yeah, I just I did a little research with biochar when I was in grad school and have a couple of papers with it. And I would say, some people think of it as a silver bullet. And there's all these different kinds of biochar and how it's created and whatnot. And I think that there's really something to it. I'm really interested personally in trying to bring some biochar into some of our composting methods or late in the composting process and see what happens, because you can have things like biochar can become a sort of like a sponge to soak up some extra nutrients and then slow release them into the system. What does that do to the soil food web? There's a lot that goes into that. But when you have the right tools and you learn how to set up some experiments and how to assess the outcomes with the soil food web as your indicator of what's going on, there's some exciting opportunities with all of these emerging ideas.
Right. Just like we talked about with the composting for municipal, a lot of it comes down into the process and having a deep understanding of what you're doing. Are you really, pushing the microbiology, or are you actually harming it? And that's kind of what you really need to look at. Okay, let's go to the next question. All right, this question is from Akari, and the question is, how do we ensure the plant is getting the correct percentage of NPK, which is the argument used by fertilizer users? Boy, I come across that all the time. Elaine, Adam, what do you guys say?
Yeah, the way you ensure that the plant gets the food that it wants is let the plant decide. And in the system that nature has put together, she actually has 3 different mechanisms of getting the right NPK into the plant, balanced properly with all the other nutrients as well, not just NPK. So when one measure is the plant puts out the exudates to control which bacteria and which fungi are going to wake up. And of course, they're going to be the bacteria and fungi that will make the enzymes to pull the nutrients the plant wants out of the sand, the silt, the clay, the rocks, the pebbles, all of that, out of the organic matter. And then when those bacteria and fungi are eaten by their predators, nutrient content inside the predators is so much less than what's in the bacteria and fungi that all that excess gets released in a plant available form right in the roots, the rhizosphere of that plant. So the plant is in control of how much of the nutrients it's going to get. What does your plant need? I think the plant knows that. So it puts out the call. It calls the pizza delivery guy and says, I want pepperoni and cheese on my extra saucy pizza. And so they make that. The pizza delivery guy delivers it to the doorstep for your plant. So that's one, probably the major way that nutrients get into the plant. We can have rhizophagy, for example, where the plant is attracting the bacteria and getting all those bacteria growing, and then it opens the doors to the tip of the root and pulls all those bacteria in. Yummy, yummy, this is so delicious. Closes the door, seals it off, and now proceeds with exudates coming from the plant, so that those bacteria give up all of the nutrients till they're almost ready to die. And then the bacteria travel up to the first roots hair, apparently, and escape out the exit doors. And then the bacteria grow. Now they come back to, where do they come back to? The tip of the root system, so they can do it again. It kind of sounds like me with, what are the rides? Yeah, roller coasters. Very good. Roller coasters. You get on roller coaster and then jump off and run to the head of the line, so you can just over and over and over again. And then there's mycorrhizal fungi, which I will let Adam make some comments about that.
Yeah, I will use mycorrhizal fungi in an example here. To answer this question, I'd like to set up two scenarios. One scenario is where you have conventional soil management, right? Industrial agriculture soils, which are broken soil food webs. And often when I went into those systems and assessed how much mycorrhizal fungi was there, it was much, much less than, say, a perennial native grassland has. The other is a soil that's restored, that has all of these components of the soil food web, especially those nutrient cycling organisms that we've talked about that are eating bacteria and fungi and releasing nutrients. I can see why an agronomy department at a university working in a broken soil system would say, well, if you're cutting hay and removing it every year, you're taking phosphorus off of the system. You're exporting phosphorus. So of course you have to put phosphorus back, right? Because you're just going to deplete it all and then you won't get any plant production. But in a system where the soil is alive and there's mycorrhizal fungi and there's all of these other processes going on, then the phosphorus is being pulled out of the parent material as well over time. The nutrients are cycling from organic matter that was in there in the past. And then you can cut hay and calculate how much phosphorus goes out with the hay and actually figure out over time how much phosphorus is coming out of the parent material. And it's a matter of matching those flows. The outflow of the nutrients and the inflow from the parent material of those nutrients. Or, you have things like legumes that can bring nitrogen back in because that's one of the most limiting factors. And so, I personally am not I'm not against somebody saying, oh, I need to go find some source of organic nutrients to put back into my farm system because my soil's not quite ready yet. I'm in transition from historic damage to a fully alive soil food web. I just don't think that they need to go to inorganic fertilizers, commercial chemical fertilizers, because those are salts, as Dr. Elaine talks about all the time. And so we're salting the earth when we're using those kind of fertilizers. But if you're composting manure from rabbits and then putting that into your soil system, you're also adding nutrients. And so it's about balancing what we're exporting in the products that we're selling and what's coming out over time. But in the case of phosphorus, we know that when people are applying that as a commercial fertilizer, it's less than 50% of what they're putting on that's even getting into the crop. And it's less than 50% of the nitrogen. So when you're using commercial fertilizers, you're fertilizing the water systems and you're causing global warming because of those transformations of nitrogen into nitrous oxide. And so you're destroying like fish populations downstream. And we've been doing it for like 100 years, and we've been watching the earth gasping for breath, right? And we can't keep doing it forever. So the status quo system of agronomy, it absolutely can't get us where we need to go to be a society, a civilization in 100 years, but the soil food web can.
Yeah, and that's something I deal with all the time, working with new clients. The modern day agronomy is actually been, so ingrained and so taught that it's actually fairly easy for farmers. It's very formulaic. I take a soil sample, I take a tissue sample, I then have an equal, an easy formula that says this many pounds of nitrogen, this many pounds of phosphorus and potassium, blah, 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 all down the road. And for farmers that are transitioning to the soil food web, it's a mindset change. It's arming them with knowledge and education about what the soil food web does and that how the nitrogen cycling works. And then I find it very useful to use some of those diagnostic tools that the conventional farmers use, like tissue analysis or plant sap analysis, to show them as we are re-inoculating that soil with a microbiome, that their dependency upon those chemical products is less. We might go from 200 units of nitrogen the first year on a massively degraded soil. And as we move into the soil food web, they may only have to put 80 units of nitrogen in that second year, and then 40 units. And then by the third or fifth year, hey, we're cycling nutrients and you don't need to put anything on the soil. And it's kind of trying to speak in their language and changing mindsets at the same time frame that is the challenge. And I think we've got the science there. I mean, we've got the knowledge and process and diagnostic tools to help them take some of their mindset that they currently had and changing it and saying, all right, now you're armed with good information. Now you can actually start to foster good soil biology. So it's a challenge, but I think it's a challenge that we can definitely meet. Okay, anything else, panelists, before we move to the next question? That was a good question. I like that one because it comes up all the time. Okay, this one's from Juan. How can you assess if you have succeeded in creating the right balance of microorganisms?
You have all the balances that you need so that the 7 overarching principles that we talk about a lot in the foundation classes, and please recognize that it's only been in the last 3 years that the last 2 of the overarching principles was really put together and put into a form that we could express the whole cycle and how it works. How many more additions are there to the list of functions that microorganisms in the soil will do for your plant, and vice versa? How much will your plant help the organisms that it relies on? It's people always want to know who's in charge. They're all in charge of some portion of the system, at some period in time. All of these organisms are controlling what's going on. So I've forgotten the question.
Oh, let me put that right back up there.
Leave it up there for a few more minutes.
Yeah, sure, no problem. It's really talking about how do we know we succeeded in creating the right balance of microorganisms?
Yep. And so your plant is going to grow without any diseases, or you're not going to get it. It's not going to get attacked by pests or parasites or anything like that. The roots are going to go down deep into the soil, and it's going to be able to reach water that's been stored in the soil since the last time you had good rainfall, typically springtime. So the plant doesn't need to be irrigated through a dry summer period. What else? You know, there's gonna be good soil structure, so water infiltrates, moves down into the soil and the root system and can take up what it needs. Otherwise, that water gets stored in the structure built by the bacteria and fungi, those open areas in the soil, because all the aggregates have been built into something, you know. So the whole list of 7 overarching principles all work together, and your plant is going to respond by being very healthy. Maximum production of whatever you planted, that plant is going to be able to express its genetic potential and give you that maximum and the highest concentrations of nutrients. There's a lot of more papers coming out all the time about how true that is, that if you get the soil functioning again with all the life that's required, then we can have plants that meet their genetic potential.
You know, for me, for me as a professional doing this kind of work, it's my microscope tells me so much information. But that aside, almost to a T, one of the things that just really blows my mind as well as my clients is at some point they recognize their environment, the ecosystem in which they're growing has changed. And it's almost like this aha moment that happens. I'm almost 100% of the time. It's just crazy. It's like, my gosh, the birds, the amount of earthworms that I'm seeing, the amount of just insect life that's not, you know, predating upon my plants, my cash crop. There's just a massive amount of insects. And it's almost as if the ecosystem has now said, aha, all right, we're in balance. Things are working very, very well. And it kind of, you know, makes my heart jump a little bit when you walk into those systems and your clients going, oh my gosh, did you see those earthworms? Did you see the birds? Did you see the insects? This thing is so much different from 3 years ago when we first started this process. And it's like, yeah, it's responded. You know, you've got that that ecosystem that's just highly functioning at this point, and it's observable. You can really, it's palpative.
It's magical.
It is. It feels that way.
I wish I could answer part of this question with saying, here's one target. If you have this many nematodes and this many protozoa and this many fungi and this many bacteria, you got a perfect soil food web. But it's just not like that. And the cycles of these things are different. And people, in a lot of ways, they need to come into relationship with their setting again and say, maybe the fungi go up and down across seasons. Maybe during a drought year, they see a lot more bacterial feeding nematodes, right? Like there's all these things that we can't know, but we can oftentimes say, you're going in the right direction. Mm-hmm. You didn't have I can't speak to this scientifically, but when I talked with Tommy, who's on our microscopy team, I asked him, when people are working on soil restoration, what organisms does he see come back in what sequence? And he says he often sees testate amoebae, the kind that have a shell around them, before he sees naked amoebae. And I'm like, well, that kind of makes sense, because if you're in a harsher environment, if you're in an environment that's not quite right for you, you need protection. And then if the environment's really, really conducive to your just being, you know, a safe, you can be a little naked amoeba. And you don't need that shell to protect you. And so I think it's about observation. It's about empowering people with the tools of observation so that they can say, this is interesting. Last time I had a drought year, this is how my soil food web shifted. And then it came back around 8 years later, we had a drought and it shifted again. Maybe that tells us something about the function of the organisms that increased versus the ones that decreased. And I mean, the sky's the limit here. We could study this for 1,000 years and keep being just in awe of nature.
And hopefully we'll still be here in 1,000 years because we start to understand this system that Mother Nature has had on this planet for the last 3.5 billion years. I think she's had a little more practice than we have.
Yes. And we, you know, mass extinction events typically aren't good things. And seems like we're rapidly heading towards one of those by our own
We're
doing.
trying to do exactly what the dinosaurs did, which was
Right.
to destroy the habitat. And oh well.
Yep. All right, let's move to the next question. This question's from Erica, and this one's teed up right for you, Adam. What happens to mycorrhizae fungi when it goes through this composting process?
I think in many cases we could see that the spores, it depends on the heat that you get to. The spores of mycorrhizal fungi can be quite sensitive to heat. But one of the misconceptions I see out there a lot in forums is that you can, like in a compost pile, mycorrhizal fungi can just reproduce and grow, or like when the compost is done. Mycorrhizal fungi need a host plant to receive food from. They have to be connected to a living plant. So if you had a compost pile that was finished and then you put sorghum or corn or something on top of it and let that grow, then you could have inoculated in some local indigenous mycorrhizal fungi and have them increase in population. But especially in the case of arbuscular mycorrhizae, which are the most common ones that relate to our food crops, they need a host plant. We've never observed a direct like saprophytic capacity for those kind of mycorrhizae to eat organic matter or something else directly. They have to have their partner. And mycorrhiza literally means like it's the symbiosis or the living together of a root and a fungi. So if you don't have a root, then that fungi is not thriving and reproducing.
It's going to hang out as a spore.
Yeah, if it survives the heating.
Up. Yeah.
Mm-hmm. Yep.
So those exudates coming from the root system are really critical.
Yeah, agreed. And mycorrhizae fungi are very important. We got to make sure that we get them into those growing systems.
On species that are supposed to be mycorrhizal.
Exactly.
There's a bunch of early successional plant species that are not mycorrhizal. They are not meant to be mycorrhizal. So if you're trying to grow these kinds of brassica, you don't really want mycorrhizal fungi in there. Yeah. So be careful on that one. You know, when do you add the spores? Make certain that that's going to be the beds that you need to grow, that you're going to grow tomatoes and potatoes and onions and garlic and all of those. Then you have another set area where you're going to grow your brassicas, your cole, your kale crops, your mustards. Just don't be inoculating them with mycorrhizal fungi because it's could be harmful.
Well, you know, it's interesting too, is that in a lot of conventional type of systems, there's always the crop rotations. You know, I got to move the crops around. And when you're really growing with soil food web, I really advise my clients to stop the crop rotation part of this, because we're going to be developing those indigenous microorganisms that are well suited for the plants you're trying to grow, and you want that to grow over and over and over again. And it's such a hard thing for a lot of folks that have come into the conventional system to say, really? What about all the disease pressure and all the other things that come, why I needed to rotate crops? We're going to let the soil food web take care of that. Have some trust in those microorganisms to be able to help us along in that sense. So,
Brian, can I ask a follow-up on that? Because what about seasonality? Like I'm used to farm systems where people are doing some kind of a winter and summer shift, and then they're also maybe interested in getting legumes in there. So like I get that crop rotations for disease and pest management and weed management aren't necessarily as critical, but what other rotations or companion kind of cycles are you finding to be true with your clients?
Well, I like, actually, one of the people I think that does impressive work in that is Rick Clark. He was in the Soil Summit, and how he has his timings down so specifically where he's able to grow multi-species crops throughout the year. And particularly a very cold climate where your seasons are fairly short. It's definitely, it can be done, but I think he's using very similar types of rotations that are going from his winter versus his spring versus his summer and so forth. And I think multi-season crop planting is fantastic. Why not? Why to take out or fallow a field when you really could have plants growing in there to really help the microorganisms in that soil.
And the reason often is that you've got to fallow it to save up the moisture. And that's just
Right.
like, oh, give me a break on this one. You have to have good soil structure so the water infiltrates and it gets below that, the level in the soil where evaporation is going to pull the water up and say goodbye to it. The root systems have to be able to get down there. Well, the water has to get down there in the first place. So you have to have structure within that soil. But water is a limiting element? No, we need to reverse people's understanding of how soil actually works.
Yeah. And I'm a huge fan of also multi-species growing systems too, especially when you're growing a perennial type of plant. If you're in an orchard or a vineyard or, something that you're going to be growing a cash crop that's, 20 years, 30 years in its life cycle, having an understory. I mean, when you go and look at natural ecosystems, there's usually an understory that's growing underneath those plants. And that again is also helping the microbiology, especially when those cash crops are in maybe a dormancy period. Well, doesn't mean your perennial plants have to be, your cover crops have to be, or cover plants have to be in a dormant cycle. So picking the right perennial understory plants is important too.
And you said something in just a few seconds ago where to me it sounded like you've been working with people that say that these orchard systems, the trees can only grow for 20 to 30 years. Mm-hmm. And then I don't know, they run out of oomph. Right. They've gotten so sick, they've been attacked exactly. For so long in the system where they have no protection. And so at 20, 30 years, you got to rip them all out and start all over again. Some of the best fruit, some of the most productive plants when we work with grapes or blueberries or even strawberries sometimes. Think about grapes. They're 450 years old. Right. And yet they're bearing just as well, if not better, than the new commercial varieties of grape because they're in healthy soil. Yeah. And so we've built up a bunch of mythologies about how plants grow, and now we have to go back and correct them so that people don't get stuck on that particular point. They're so surprised when their plants are still alive after 30 years.
It blows my mind. When you look at some of these systems, you're like, walnuts, you're taking them out after 20 years.
They're just getting good.
Exactly. Yeah. So I think just like you said, Elaine, it's because of the way those plants have been managed. They are just beat. There's not a good immune system with those plants, and I think for sure that's going to change when we start getting a lot more acreage underneath, growing with biology, that we'll look at orchards and say, well, it's 20 years. Why would I take these plants out? Mm-hmm. Yep. Their 20th year was more productive than the 19th year, which is typically not the case. There's usually a curve. And then as they get it to those older ages, the conventional systems, they start to crash. And that's why they pull them out.
Yep. So. And it just seems such a pity to be so oblivious to the pain of those plants.
Right. Agreed. Okay, let's go ahead and move to another question. This question is from Rodolfo. And the water to be used for compost piles, if it is tap drinking water, is there a recommended procedure to make it more compatible with compost microorganisms, e.g. aeration/sunlight exposure ?
Well, the real problem with drinking water is it comes with a load of typically chlorine or chloramine. And both of those are highly detrimental to the microorganisms in the soil. It will kill a large proportion of that community. So really you want to add something to that tap water. And people will always say, well, why are those toxic things in my drinking water? It's because when the water leaves the water treatment plant, it is sterile water or very low levels of anything. But as they go through these pipes, the pipes have gotten biofilm growing on the inner surfaces. And so these chemicals are additions to remove that biofilm, to prevent the biofilm from getting any worse. So if you live really close to the water treatment plant, you're going to get a huge dose of the chlorine or chloramine. A little bit further out, it lessens every foot that you go. If you happen to live on the very end of that water pipe, there will be almost no chlorine or chloramine left in that water. So you got good drinking water. Don't worry about getting rid of the chlorine and the chloramine. But if you're anywhere in the in-between, what position does your house have on the water pipe? Nobody knows. They just totally clueless. So to be on the safe side, you want to be putting about 1 drop of a good humic acid into your water per gallon. And so you can just have a little container of humic acid that slowly, a Venturi system, slowly drips that 1 drop of humic acid per every gallon of water that's passing by coming from the main faucet. And then you don't have that problem of losing a huge number of your organisms that you've worked hard to get back into your soil or back into your compost. And other things can be used like citric acid, all of those organic acids. The thing about the humic acid is it turns the color of the water just the slightest bit tan. So you can usually tell if you're putting enough humic acid in because you get to see the color change. Anything else that just is clear and it looks like water, how do you know if you've added enough?
Water quality is an issue, and I look at the chlorine and chloramine is kind of a necessary evil in our water delivery system, especially for municipalities and cities and so forth. You don't want Legionnaire's disease or all those other things that you can get if you don't.
E. coli, Salmonella, Shigella.
Yeah, but in the agricultural space where we don't usually use municipal water, there's still water quality issues. One of the challenges we're running into now is that a lot of the easy access groundwater is gone. And so there's a lot of farmers that are drilling very, very deep wells, and they're pulling up water that's high in a lot of bicarbonates. It's very, very salty water, and we have to be able to deal with that as well. So it's not necessarily a chlorine or chloramine type of issue, but it's a salt contamination issue. And as Elaine mentioned earlier, salts can be the enemy of the microbiology world. So now you have to treat that, and it's an expensive process, but it's something you have to pay attention to. When you go into any kind of farming system, you just don't look at just the soil or the compost you're using. You have to take in a whole litany of things, like the water that you're using, what's the equipment and the sprayers and the pumps and all those other kind of things. So yeah. Adam, anything you want to add?
We got it covered. Elaine got it covered.
Exactly. Good job. All right, let's move on to another question here. So this question is from Teddy. And Teddy asks, can't turning compost piles actually disrupt the fungal networks?
Sure it does, but it's a kind of what's the worst case scenario. If you don't turn the pile, then you're going to get areas where you have just this kind of fungus growing at one species, king of the hill. Over here, you've got some bacteria. Over here, you've got, and your pile is just these little pockets. And we want to have a nice uniform application of this massive diversity. We want to have everything being inoculated, and instead of just 1 or 2 species getting through. So you do want to turn the pile. You also get little places where just one type of food is here, 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 and you need to break those pockets up so you have a good uniform product.
Absolutely, yeah. And we're not advocating that people just blast these piles with, some kind of equipment or something. It's like with a pitchfork, some good, muscle power, or with the specialized piece of equipment. So yeah, the mixing is incredibly important.
I always look at it as you are turning the minimal amount of times to ensure that you still have aerobic composting occurring. And then once you've gone through so much of the compost, you've built enough structure, and by this time frame, you probably also consume most of the high energy products out of there. Now there's no longer a need to turn. And my experience is that as I let my compost mature, I'll start to see those fungal networks continue to grow and grow and grow and grow. Yeah, again, it's kind of that necessary evil thing. Need to do it because I want to make sure it's aerobic, and if I don't, it's gonna be a problem. Okay, anything else you guys want to add? All right, let's jump to another question. I think we got one more question here, and then we'll probably be just wrapping up the webinar here. So this question's from John, and this is for Dr. Elaine. I have some mixed grass bales and cornstalk bales I'm interested in composting and using for fertilizer. What is the best method in creating the environment required for my microbiology to break down and create the proper compost required to spread on my farm?
What's the best method? It's to use the soil food web approach in making actual compost. So we want to make certain that you're getting quality materials that should have all of the really beneficial organisms on them that they possibly can. No pesticides, no inorganic fertilizers, because you just killed most of everything. And if you bring those materials into your compost pile, it's going to take its toll right at the beginning, right where you want all that diversity to be present. Now, you get a couple handfuls of not so wonderful stuff. Okay, it's going to be able to, your pile's going to be able to overcome that. But if you're putting in, 10% of the pile that has horrible toxic chemicals in it, like in the manure, for example, then usually you can't even generate the heat required to kill the pathogens and pests and problem organisms in there. So make sure your starting materials are good. You can even test them in very small piles. You can add, different combinations of the woody, the green, and the high nitrogen. So that's the best method, is good organisms going into the pile, and then you've got to make certain that the pile stays watered, 50% moisture, please, and that's free water that we're talking about, not gravimetric water. So you want to make certain that anytime the pile gets nice and toasty warm, you've been above 131 degrees Fahrenheit for 3 days, and yeah, we've got all the rules in there, and we teach you all the rules. And I could go on for the next hour and a half just talking about how to build a pile. So we slowly but surely teach you how to do this step by step. And, most people get really excited because it's easy. It's really not that much work. We can make finished compost in 21 days and make sure that it is biologically complete, that it meets our minimum levels for the microorganisms within that pile, because you will be able to see benefit to the plant growth. Now, if you want to see the maximum best response, well, then we got get down a little bit more down and dirty and make sure we get all of the different factors. I know I kind of buzzed through all of those factors early on because I didn't want to take over the talk.
Right. Well, and I think you hit it right on. There's a recipe, right? There's a way that we go about in assessing the materials that we're going to use, see where they fit into the high nitrogen or the green or the woody process. We build piles, we get data, we look at that data and say, all right, we need to modify this a little bit. What I thought was high nitrogen was maybe not quite high nitrogen. It was high nitrogen and green, really fits in that category. And then you kind of refine that recipe until you lock on to a really good composting process. And the other thing I think you mentioned too is that there are multiple composting methodologies, and they have their pros and cons. There's static composting where you're not turning it. It's very, very low management. You have the thermophilic composting, which was what you were talking about, Elaine, where we're managing time, temperature, and moisture. So we can go through a very intensive composting process and have good compost after 21 to 28 days. There's vermicomposting. There's things like the Johnson-Su bioreactors, so there are methodologies that are out there that have their pros and cons, and you kind of fit with what are you trying to accomplish, what materials do you have, what's your time frame that or labor that you can put into this, and then you pick and choose the right methodology from there.
And we do add those things into the compost class. We talk about doing all these different kinds of composting. So it's just that the thermophilic version is almost without a doubt going to give you a product that you're going to be very happy with. You're going to have the organisms. You can look at them through the microscope. When you deal with static piles, you have the problem of little patches that are one thing or another. And because in a static pile, you don't turn it, you don't mix it. The middle of the pile can be very different from the outside part of the pile. When you're dealing with worm composting, you can have some great compost from that. It's just that you can't make massive quantities of it very easily, whereas with the thermophilic pile, you can make mass quantities of really good compost fairly easily.
Yeah. Adam, anything else you want to add before we close out the question?
Yeah, I think that the biggest danger would be if these, especially mixed grass bales, had a bunch of herbicide residue or something on them. You really got it. The devil's in the details, right? It always is. So understanding what the inputs are is, as critical in some ways as getting the balance between different types of materials right in terms of having a thriving composting process.
Yeah. Okay, well, I think that's going to conclude our Q&A section. I'm going to go back into slideshow mode here just so that we can see. And what I'd just like to mention before we close today's webinar is that we are in our April promotion. So this is the Springboard Plus offer. So typically it's $5,000 for our foundation course classes. And again, we're 47% off, so at $2,900. And again, you get those two courses, the permaculture and the DD Pure House's classes as well.
Really good classes.
Yeah.
It's well worth it.
Yeah. Exactly. I agree. And I do want to also tee up the rest of the webinar series. So we've done 1 and 2. And again, you'll be able to watch those replays if you weren't able to watch webinar 1, or you want to rewatch this one. There's a lot of great information that we covered here today. But webinar 3, which is going to be at 9 a.m. Pacific timeframe on Wednesday, April 20th, we're going to have Ray Archuleta. And I know a lot of people know about Ray Archuleta. He is definitely a very great leader in the regenerative agricultural space, and he's a fantastic orator. I just actually love hearing Ray talk. He's a fun guy to listen to. So I definitely recommend watching that one. And then our webinar 4, which is careers in soil regeneration. So talking to folks that are actually out there, helping their clients transition from conventional or conventional organic to farming with biology. And really, how does that process work, successes that they've had, and things like that. So, and that one will be at 11 a.m. Pacific timeframe on Wednesday, April 27th. So we hope to see you folks there. And we also mentioned our January series. You can go back and rewatch those if you hadn't had a chance to look at that. There's some great information in the January series as well. Okay, at this point, I'd like to give a big shout out and thanks to the support staff that set up these webinars, help us make this run smoothly behind the scenes. It is really a team of people that it takes to put these series on, and I want to thank everybody who's working with us to make these things a success. I want to thank today's panelists. So Adam, always, it's been great having you as part of the Soil Food Web School. You bring such great knowledge to the program. I really appreciate you having on our webinar today. And big thanks out to Dr. Elaine Ingham. She's really, been the driving force in the Soil Food Web, and really has pushed, I think, most of us and inspired us to do this kind of work. So thank you very much, Adam, and thank you very much, Elaine.
Well, thank you, Brian, for being the host for this and keeping us on time and moving along. Wouldn't be able to do this without you and Adam to help, and of course, all the behind the scenes folks. Thank you so much.
Absolutely. I had a
Thank you, thank you.
blast.
It's great. It was fun. Okay, at this point, we're going to end the webinar. So see you folks at webinar 3.
Yep, take care. Ciao.
Don't forget to click that like button, subscribe to stay new videos.